So let's begin our today's discussion. First, we will start with the introduction of genetics. So what is genetics? Genetics is a branch of biology that deals with heredity and variation of organism. So yes, it deals with heredity, and this term you are actually know this term from your class 10 or 9, 10 onwards. So what is the meaning of heredity? Heredity means the movement of genetic organism from generation after generation. And now, what is the genetic component? Genetic component we define as chromosome or genes. So what is chromosome? Chromosome are the carriers of heredity or information. So chromosome is a specific part which is actually visible in the metaphase stage of the cell cycle. So this is actually visible in the metaphase stage of the cell division, and it's an arrangement of nucleotides in DNA. We will learn the nucleotide arrangements and everything in our subsequent lectures. So let's begin with the with some terminologies like chromosomes and homologous chromosomes. Chromosomes occur in pairs, and we call these pairs are the homologous chromosome. Example of this is two chromosome, as say chromosome number 1 of human. So these are the two homologous chromosome, and the homologous chromosome actually contain the same gene in the same position. Okay, so these two chromosome actually comes from the fertilization from two parents. So one chromosome comes from father, another comes from mother. So that we need to know, that we need to understand. That is why we are the diploid organisms. So new combination of genes occur in sexual reproduction. Yes, sexual reproduction develops new combination of genetic feature which comes both from both the parents. So now, who is Gregor Johann Mendel? Gregor Johann Mendel is an Austrian monk born in what is now called Czech Republic in the year 1822. He, he is a son of a farmer, and he studied theology and was ordinated in a priest in St. Augustine. Went to the University of Vienna, where he studied botany and learned the scientific methods, different scientific methods. He worked with pure line of peas for 8 years. And prior to Mendel, heredity was regarded as a blending. And from the Mendel experiments onwards, we are able to know that the term genetics, pro blending process, and offspring were essentially a dilution of the different parental characters. But after Mendel's experiments, we are able to understand that offspring are the mixture of the two characters from the parental origin, not the dilution of characters. So Mendel actually experiment with the pea plants. He tried with the other plants, but he never succeed with the other plants. But whatever his experiments, he actually conducted all of his experiments with the pea plants. Now Mendel actually uh, experiments or selected 7 different traits or characters of pea plants. So the 7 different characters are round and wrinkled, yellow and green. These are the opposite characters. Sorry. So purple and white flower, green and yellow unripe pod, inflated and pinched ripe pods, axial flower and the terminal flowers, long plant and short plant. So these are the 7 different characters of Mendel pea plants. So now he published his experimental papers in experiments in plant hybridization in 1866, in which he established that three principles of inheritance, which we are going to discuss in our classes. So he tried to repeat his work in another plant, but it didn't work because the plant reproduced asexually. So that means whatever the basis of Mendelian genetics, that basis is based on the sexual reproduction. His work was largely ignored for 34 years until three independent botanists rediscovered Mendel's work. So now Mendel was the first biologist to use mathematics to explain his result quantitatively. Before that, until and unless we use mathematics or statistics in our in biology, it only gives us qualitative results. But the Mendel was the first scientist who used the mathematical approaches in biological fields and got his results. So Mendel predicted the concept of gene. This is a very important uh, part of the discovery. From his experiments, we are able to understand the concept of genes, that genes occur in pair. This is also a very important part. And from there, homologous and heterologous concepts come. That one gene of each pair is present in the gamete. That is also very important. So that determines the haploid from the diploid organism. So now we need to know some terminologies which actually deals with the genetics. So first is the gene. Gene is the unit of heredity that we all know, but that is not a complete definition. Gene is a section of DNA sequence which encode a single protein. Now it's a complete definition. So what is gene? Gene is a heredity, unit of heredity, and a section of DNA sequence that encodes a single protein. Now it's come the genome. Genome is the entire set of gene of an organism. Now it comes the allele. Allele, just let me explain what is genome once again. Genome is the entire set of gene of an organism. Like that, we have 23 pair of gene. That means 22 pair of homologous chromosomes and one pair of sex chromosomes. In a complete way, it's a 23 pair of gene. That means there is a 46 number of gene. So whatever the total gene amount which is present in this total 46 number of gene are actually cumulatively called genome. That is the entire set of gene of an organism. Now it comes the allele, two genes that occupy the same position on homologous chromosome and that cover the same trait. So allele are the two genes that occupy the same position on the homologous chromosome and that cover the same trait. Like they have the similar characters. Let's just back a few slides. Just take an example of this slide. These are two homologous chromosomes. These purple genes and these light purple genes are present in the same position of the homologous chromosome, like the other one, this one, these green genes. So these are called alleles. So now allele are the two genes that occupy the same position on the homologous chromosome. And now the locus, the position. The position is the locus. So the fixed location of a strand of DNA where the gene on one of its allele is located. That is called the locus. So now it's a homozygous. So homozygous means having identical gene of a particular characteristics. Heterozygous means having two different genes of particular character. That means homozygous, we can consider capital T as a homozygous, and we can consider capital T and small t as a heterozygous condition. So what is dominant? Dominant are the allele of a gene that mask or suppresses the expression of an alternate allele. The trait appears in the heterozygous condition. So dominant means dominant is a particular characteristics of a gene which is expressed, and if a dominant allele is present between a heterozygous uh, characteristics, then in this particular case capital T is the dominant and small t is the recessive. So always capital T express its character. Now is the recessive. Recessive is an allele that is masked by the dominant allele. That is why when capital T is present, the characteristics of small t is getting masked. That is why small t is known as the recessive allele. Okay, so the characteristics of recessive allele is always masked when it is present in a heterozygous condition. But the characteristics of recessive allele will only express when it present in a homozygous condition. Now, what is genotype? Genotype is the genetic makeup of an organism. If we consider these two example, in both of these case, capital T is a dominant one. So in both of these organism, it will always express the characteristics of capital T. Fine. But genetically, if we consider the genetic makeup, these two are different. This is the homozygous condition and this is the heterozygous condition. So that is why the genetic makeup of an organism is called the genotype, and the physical appearance of an organism is called the phenotype. Fine. So now we are going to the monohybrid cross, a genetic cross involving a single pair of gene. This is a single pair of gene. Like single pair means gene having a single character. If we consider the cross between these two are actually encode a same character, that is the length of the plant. Okay, so that means a genetic cross involving a single pair of gene that is differed by a single trait. Now P is the parental generation, F1 is the first filial generation, that is the offspring from the genetic cross, and the F2 is the second filial generation of genetic cross. So now we will directly go into the monohybrid crosses. So monohybrid cross means parents differ by a single trait, and that cross between parents. So this is the long tall plant and this is the short plant. So we call it a capital T and it's a small t. So crossing two P plant that differ in the stem size, one tall and one short. Capital T is for a tall and small t is for the dwarf or short. So capital TT is the homozygous tall plant. Small tt is a homozygous dwarf plant, and if we cross the capital T that is a homozygous tall with the homozygous short, we will get this kind of plants with the stem length. Let's check out the cross between the capital T and small t. That means a tall plant and dwarf plant. So the cross between the tall plant and dwarf plant resulting in a all tall plants. So what are the possible gametes produced from this tall plant? The possible gametes are capital T. And this small dwarf plant, it's a small t. And the combination of any of the combination results the same trait. I am just showing you the different kinds of combination. So any of the combination will produce the same trait, that is the heterozygous tall plant, and we call it a F1 generation. So this is the Punnett square. Punnett square is a useful tool in the genetic crosses. So for monohybrid cross, we need a square that is divided by 4. We need a square of 4 boxes. So it's look like a window pane. We use Punnett square to predict the genotype and phenotype of the offspring. That is very important. We are able to predict the genotypes. We will look at an example just after this slide. So this is how we use Punnett square. Step 1: determine the genotype of the parent organism and write down the cross. This is the parent organism. This two. And if we write the cross here, we are actually able to take two different T and the small t. So we can write capital T, capital T. These are the gametes actually, which we write in the top and the side of the square. And this is the small t, small t. So the cross between these two results the capital T, small t. When this capital T and this small t crosses, this results the same feature. So by using Punnett square, we are actually able to split the letters of the genotype for each Punnett square and put them outside the P square. Just like this, we have put the gamete letters in the outside of the boxes. Determine the possible genotype of the offspring by filling in the P square and summarize the results like this. Whatever I have written in the earlier slide, here it is being shown. And how we are able to predict the genotype here? Here we are able to see that every plant, every offspring have a capital T, small t. So the genotype is capital T, small t. And you need to write the heterozygous tall plant here. And the phenotype is also the 100% phenotype, and which represents the tall plant, because a capital T gene is present in every boxes, in every offspring, right? And the capital T is a dominant gene, and whenever a dominant gene is present with a recessive gene, always the expression of the dominant gene occurs. So now we are going to see the monohybrid cross of the F2 generation. So F2 generation starts with the the crosses self-fertilization of the F1 progeny. So in this case, if you let the F1 generation self-fertilize, the next monohybrid cross would be capital T, and capital T, small t. So in a similar way, we have to take this capital T and this small t here outside the boxes, and this capital T and this small t here in the outside of the boxes. So, and we get in cross between these two results, capital T, cross between these capital T, small t result this, cross between these capital T, small t result this, and cross between this small t result this. So here, from here, we are able to get one capital T plant, two capital T, small t plant, and one small t dwarf plant. And we get the genotypic ratio of 1:2:1. And similarly, we will get actually three tall plant and one dwarf plant. So the phenotypic ratio will be 3:1. So the secrets of Punnett square. It's a it determines the gamete of each parent. We have to determine the gamete of each parent by splitting the genotype of each parent. Now, if this is your cross, the gametes will be capital T, and small t. I have already shown this to you. So once we have the gamete, we have to just put this gamete outside of the boxes, and then we have to just go for the cross between each and every possible gamete. Fine, and we get the possible results. So if either parents is homozygous, we will get 100% genotype of homozygous plants, and 100% phenotype of the tall plant. So understand. So now, if we have one homozygous and heterozygous like this, we will get the. If we can use only one box to determine this. There is no need to write these whole Punnett square, because whatever the gametes, that gametes should comes each and every gamete has the same properties. So if but if we have a cross with different kinds of gametes, like one heterozygous with the one homozygous, this one is the heterozygous and this one is the homozygous, then we have to use the then we can still use the shortcut, because in this case only one gamete is being produced, and in this case the two gametes are produced. The two gametes are capital T and small t, and from here we get the small t, and we will get a ratio of capital T small t, and from here small t. So these two. So in this case, we will get 50% of the capital T small t, that is the heterozygous tall, and 50% of the small t, that is a homozygous dwarf. And the phenotype is also the same. So here we can take another example of the flower color. So here we can use purple color. Purple is the dominant one, and small p is the recessive one. So if you cross a homozygous purple with a homozygous white plant, we can get this purple flower with a dominant character. That is a heterozygous purple flower. So all flowers are purple. So thank you. This is the end of our today's class, and we will continue our Mendelian genetics class, Mendelian genetics topic actually, in our subsequent classes. Bye.